To measure stress in the brain, we have to do mainly two things. First, we have to elicit stress in a suitable stress test, and we have to try to obtain information directly during the stress experience. We are doing this by performing a stress test in the fMRI scanner and obtaining a lot of parallel signals. Stress not only elicits a brain reaction, but stress elicits also reactions in our body systems like skin conductance changes, heart rate changes, breathing changes, changes of stress hormones that are released into the blood. All these elements can be measured during our what's called multimodal stress test. To directly measure what the brain is doing during the stress experience, we are using a kind of trick in our stress experiment. We first ask the participants to perform a cognitive task to activate the brain generally. Then we put this task under stress, for example, by giving aversive feedback, by exerting time pressure, by creating a very uncomfortable social atmosphere, like in the TRIA social stress test. And then later we can subtract these two conditions in an attempt to isolate what is the brain doing to mount this additional stress that we put on it. During our imaging stress test, we try to obtain as many signals of the stress response as possible. One important layer is the sympathetic nervous system response. It occurs very fast, and we try to obtain it by measuring heart rate and skin conductance. On the other hand, our organism adapts to stress more in the long run by releasing stress hormones. And these stress hormones we can measure by using such probes repeatedly during the stress test. The participants have to chew on this, and we then analyze their saliva to get their levels of cortisol and other stress hormones. So our stress test that we designed to study stress consists of several sections. We are starting with a resting period during which the participant is not asked to perform any specific tasks. We then get into the pre-stress phase, in which participants have to do an easy cognitive calculation task, interrupted by small sections of breaks. We then exert time pressure and other aversive feedback to really put our participants under subjective stress. And after that, we take back these stressors and switch back to the normal, easy cognitive task. After these 3 blocks, we have a recovery period, and finally, we will obtain another resting state scan of the brain. During this whole process, we measure stress reactions at different levels. First, we get signals from the sympathetic system. We get hormone levels, for example, cortisol, and before and after the stressor, we also ask our participants how they feel during the stress test. From our previous analysis, we can see that our stress test really elicits stress reactions. We can see pulse rates shoot up compared to a control group. We can see that cortisol levels increase as we would expect with a certain delay after the stress intervention. And we also can see that negative feelings of stress and time pressure are also clearly elicited. To really be sure that we extract information from our images that relate to stress itself, to the regulation of the stress response, we combine our image data with other measurements. For example, the measurements of the sympathetic nervous system or our cortisol measurements. By this step, we can ensure that our results become more specific to what we actually want to study, which is the stress response of the brain.